Awesome. Okay, so hi everybody. My name is Julie Larson. I'm a postdoctoral research ecologist with the USDA Agricultural Research Service in Burns, Oregon. And I want to talk today about this idea of using seed and seedling functional traits as a tool for decision-making in seed-based restoration. These are some ideas I've been talking about with my co-author, Stella Copeland, and some of the collaborators that you see up here. We just submitted a paper laying out some of these ideas that you're going to see in this talk, so hopefully that'll be out soon. But I want to start today by bringing you into the systems where we're working, the western drylands of the US, the Great Basin and the Colorado Plateau, which are home to a diverse range of cool and warm season grasses and shrubs and forbs. And when we think about desirable restoration species in these systems, there's a bit of a gradient, from species that are found, a little bit more in the cooler Great Basin to the warmer Colorado Plateau. But these are just 6 of the 50 restoration species that we're collecting trait data for, to give you an idea of the diversity of species that span these dryland systems. And these systems, of course, also have some major exotic species that we're collecting trait data for. 2 that we're studying do an excellent job of competing with natives, and 2 cool season grasses, cheatgrass and crested wheatgrass. And when we think about the major challenges to restoring these systems, especially after major disturbances like fire, I think there's no doubt that the limited availability of seed is challenge number 1. However, I know a lot of folks at this conference are working on this problem. So in this talk, we're going to think optimistically, and we're going to skip ahead to challenge number 2, which is, once we have these native seed sources available, how can we use them more effectively and efficiently in seed-based restoration? And this is also a really critical question, especially in dryland systems where seed establishment is notoriously low. So a recent global synthesis from restoration experiments in dryland systems found that over half the times a species was sown, no establishment, 0 establishment was detected in subsequent monitoring. And this is a problem because we're thinking more closely about which native species we want to restore, but we also know that limited establishment can make it difficult to achieve these targets. So evidence suggests that initial seeding success can be limited very early on, based on how successful seed germination is and how seedlings survive potential hazards around the time of emergence and establishment. We also know that each species has its own recruitment niche, a unique set of conditions where its seeds can successfully germinate, emerge, and establish. And I think if we could characterize the recruitment niches of our species in a way that's accessible, we'd have a basis to strategize seeding in a way that overcomes some of these establishment limitations. So I'm going to suggest, as a working hypothesis, that seed and seedling traits could be used to understand recruitment niche differences among diverse species, and also to address these challenges in restoration. And we're going to demonstrate these ideas using trait data for our 50 dryland species. But first, I want to walk you through the trait data that we're currently collecting. So a trait could be any character of a species, right? But I think mostly about functional traits, which are attributes that directly impact a plant or a seed's performance in the environment. So for example, differences in seed size are critical because smaller seeds, they tend to persist longer in the seed bank, but they also hold fewer nutritional resources for a seedling that might be going through resource stress or limitation. And differences in germination timing, also influence whether seeds all germinate immediately or whether they delay under certain conditions, which influences the risks that seedlings face during recruitment. And even at this really young stage, these little teeny baby plants, they have different traits that can impact their performance. And we're growing species in this clear agar so that we can actually see and track traits related to seedling growth, root investment and development, and seedling tissue toughness, traits that might also explain differences in drought or freezing tolerance, which we're collecting for these 50 species right now. But today, we're only going to look at a small subset of these germination timing traits. They already pack a ton of information, which you'll see in a little bit. And we'll use these traits First, to just explore some of the variation in species recruitment strategies, whether there are interesting differences among plant functional groups, different commercial varieties of the same species, and common exotic species. And then we'll explore how we might use these traits to address some restoration challenges. So let's start with some seed dormancy and light response data. If anyone studied these things, you'll know there are probably a million ways you could quantify both of these metrics, and catch me later if you want to chat about it. But we boiled it down to two metrics here. So our dormancy metric on the x-axis is meant to capture variation in the extent of dormancy, and it's simply the percentage of viable seeds that germinated in, oh, I hear the bell. It's the percentage of viable seeds that germinated in the first 4 weeks without any dormancy breaking treatments. And I realize it's a little bit backwards, but species with lower dormancy levels, are on the left, and species with higher dormancy, with more dormancy are on the left, and higher dormancy on the right. And light response, our light response metric, is simply the difference in percent germination that we observed between light and dark conditions under otherwise optimal conditions. So each of these points is one of our 50 species, and just to demonstrate, these 6 points are the species that I showed you at the beginning that I highlighted earlier. And along the x-axis, we can see just how much variation there is in the extent of seed dormancy across our species, ranging from desert senna on the left that was completely dormant to basin wild rye on the right, where almost 100% of seeds germinated without any dormancy breaking treatment. And I should note, for the species assessed as having higher dormancy, we did subsequently apply dormancy breaking treatments to test this. These included scarification, where we broke the seed's seed coat. We broke the seed coat to allow water absorption. To allow water absorption and embryo expansion, and also stratification, where seeds were exposed to cool, moist conditions to break physiological dormancy. Requiring scarification and stratification, these are two different mechanisms to delay germination and avert or spread risk in the field. So the symbols that you see now reflect what ended up being the best dormancy breaking treatment for each species. And you can see that high dormancy levels are attributed to many different mechanisms, which I think could even widen the spread even more of field germination times that we see when we seed these species. Now, in terms of seed light sensitivity, you can see two things. First, there's not as much spread, right? Most species had relatively little light sensitivity. Though a few had some more extreme requirements, especially for dark conditions, the two grasses down here, Lamus and Elamus. This could impact how we choose to seed different species, whether it's critical to bury or cover them, or whether they need to be on the surface. I always say knowledge is power, right? We have this information, we can use it in a lot of different ways. So these traits, speaking of that, could also be useful to understand how exotic species are functioning and whether there are any how native strategies compare to that, right? So when we look at cheatgrass and crested wheatgrass, you can see here they're among the least dormant species and with relatively little light sensitivity. And when we look at the data this way, we can also identify, which natives are the most similar. So when we color these points by functional group, you can see that some of the species that are most similar to our exotics include other cool season grasses, but also a couple of forbs in purple, some warm season grasses in blue, and even a legume out here that has physical dormancy, but still germinated pretty well even when we didn't break the seed coat. So you can see that one of the things to note is there's wide variation within functional groups. So the colors are all spread out. And to me, this suggests that it may not be easy to infer or generalize species traits based on other species that we think are similar, because they might have a lot of different things going on. We also saw that when I'll show the next one, when we included two commercial varieties of the same species, they could also differ. Like we see for Lamus cinereus basin wild rye here, we had one variety that was not light sensitive at all, and one that was fairly light sensitive. So finally, I just want to point out that seed light sensitivity and dormancy level weren't really related, suggesting that these are two separate mechanisms of germination delay. And a third possible mechanism that's important in our system is temperature sensitivity, which can also delay germination in the field. So we track germination rates at 3 different temperatures and have quantified sensitivity in 2 ways. So first, on the x-axis is the minimum coldest temperature at which at least 10% of the seeds were able to germinate. And on the y-axis, we estimated just the raw difference between the total germination we saw at 18 degrees Celsius versus 6 degrees Celsius colder condition. And again, we saw pretty wide variation in temperature sensitivity. So you can see here, this is a cool season grass. Again, our Great Basin wild rye could start germinating at temperatures as cold as 2 degrees Celsius, versus Bouteloua gracilis, which is a warm season grass, needs at least 7 degrees Celsius to start germinating. And when we look at functional group patterns, we actually see a meaningful split this time, right, between our cool season grasses in light blue and warm season grasses in dark blue. And we know that warm season grasses are adapted photosynthetically to handle warm conditions, but we see it starts at the seed. They're delaying germination until conditions get warmer, which is really cool. And finally, when we look at multiple varieties of the same species, we again see that there's this potential for variation within species. We've been hearing a lot about that at this conference. It's not a surprise to anyone, but it's important to remember that depending on what seed source you get, traits can be different. Okay, so now in one figure, you can sort of see the 3 trait dimensions we've talked about, light, dormancy, and temperature sensitivity. And now's the part where we brainstorm how we could use this data to address some restoration challenges. So let's start with this first challenge of asking whether traits can help us select forward-looking restoration species. What do I mean by forward-looking? Well, in many systems, including the Great Basin, climate forecasts suggest that more frequent and extreme weather events are coming our way, becoming the norm. So a common forward-thinking goal is to restore plant communities that have some buffering against environmental uncertainty or variability. And buckle your seatbelts, but I'm going to suggest that we could design a seed mix to select species that have diverse dormancy strategies and maybe even emphasizing species that have stronger dormancy levels. Because in my mind, I think we only get to go out and seed once. And what if we could spread the risk, and reduce the chance that all seeds germinate immediately after seeding? Spread the risk in the near term and establish species that might establish more resilient seed banks in the long term. Speaking of seed banks, let's look at challenge 2, anticipating recovery. This is a major challenge because a lot of work has suggested that passive recovery of systems after a disturbance like fire can sometimes be decent. Seeding doesn't necessarily always improve outcomes. Like, what if we could use seed traits to predict what was already stored in our soil seed banks? Now, I believe light sensitivity probably is a useful trait here, but I'm going to focus on 2 traits that I actually have some data for, seed mass and life history. And what you're seeing here, we got to hurry up. Okay, so what you're seeing here are data from that I've collected from a Colorado grassland, a separate system, showing about 100 different species here, and how common they were in the vegetation versus the seed bank on the y-axis. And what you see is that seed bank prevalence on the y-axis can be estimated fairly well based on seed mass and life history, in part because small-seeded annual biennial plants can make a lot more seed, and because small seeds are more persistent in the soil seed bank. And we'll skip ahead to this third challenge. Finally, we can use traits to match seeds to appropriate seeding approaches. So for example, in a scenario where a site has some big pockets of exotic cover, we might seed a strategic subset of our seed mix selected based on similarity to exotic species. So if these are our 2 exotic species in red, we can identify species that have similarly low dormancy levels, light sensitivity, and temperature sensitivity, and maybe even identify some new species beyond the typical players. Here you see in one of our species is Linum lewisii, blue flax. It's not always used to compete with cheatgrass, but maybe it could be. So I'm going to wrap things up. And I just want to acknowledge that At the moment, this really represents a set of ideas. There's so much more data that we need to collect and testing that we need to do to see if we can make decisions based on these traits, how we design seed mixes and use them in the field. Moving forward, I wonder whether we can leverage existing systems to collect more and relevant seed and seedling trait data. So for example, by creating partnerships between researchers and seed testing facilities that are already measuring, seed germination and seedling vigor, or with plant materials agencies that are already measuring traits in new plant materials. And when individuals or organizations collect trait data, can we make it more accessible? For example, by establishing regional databases that are accessible to users. We have a few global seed trait databases in development, including one that I'm working on, SeedArc. But these may be overwhelmingly big or complex to use in local restoration projects. And finally, this is the big one. We actually need to test seed mixes that are designed on the basis of traits to verify that we can get stronger establishment and better outcomes with this kind of approach. I think this is a natural area for researchers to partner with agencies that are already doing restoration projects like BLM, but we have to keep building connections and get some planning going in advance so that we're ready to test things out when a disturbance occurs. So with that, I will leave you with our species list for anyone that works in this system. If you want to talk about seeing data or sharing data or anything else, come find me. And thank you again for listening to some ideas.
We have 3 minutes for questions.
There's a lot of data. I know, it was like, I only showed a couple of data figures, but there's so much information packed in there, right? I knew Todd was going to ask a question, especially because his face looked so confused.
Julie, good talk. Of the traits you didn't show,
I didn't.
Are there any sort of winners that are outside that norm, seed mass, like sensitivities, or any other ones that you really see as a magical trait that could add some?
Right. Yeah, so the question is, I only showed like a bunch of germination-related traits today, but we're collecting right now a bunch of different seedling traits. And I think the key, too, is someone had mentioned earlier, maybe Kevin Bladdock, but we're monitoring seedling growth at multiple temperatures because when we're trying to predict, how well seedlings are going to grow and establish, that process, too, is super temperature dependent, and different species have different temperature sensitivities. So we're going to be tracking that, and then we maybe can use that in models. How quickly the roots are extending, and at different temperatures. Todd was asking is there going to be like a magic bullet trait or something that I'm really excited about? And I don't know the answer to that yet. And one of the things I'm waiting for is like right now, our technician back in Burns is trialing out how we're going to do these freezing experiments where we get the seedlings going for about a week and that's it, and then we freeze them. And there actually are, there's differences in which different species can take different degrees of freezing, and we'll be able to quantify that. Same thing with drought tolerance. What I'm waiting to see is if any of the traits that we're collecting on how roots develop and how leaves develop are linked to that survival capacity. Because we know freezing is a major mortality factor in our system, and drought is too. Those are two of the big ones. So if we have that, even that information on itself, I'm really excited about. So.
I'm interested in your suggestion about leaning towards species before dormancy, before species before dormancy. The wetland species I work with, I think we're actually maybe approaching thresholds where the winter temperatures are actually not going to be sufficient to break the dormancy. I'm curious in your system if that may be a concern as well.
I have been thinking about that. I don't know. I'm not a climate expert, but definitely find folks in the Great Basin who would know that. I don't think it's an immediate concern, but it could be. And like I said, knowledge is power. And with this kind of approach, at least we know, right? So one of the things that I didn't mention is that when we were stratifying species, we had some species that probably within a week or two of stratification at 3C started germinating. Like they just needed a little bit. And then we had some species that after 6 weeks, nothing. After 8 weeks, nothing. I think we finally let them go for like 11 weeks, and then they finally was enough, right? And so this is why I'm like, we need to collect the information so we know. I don't know if it's going to get warmer. Well, I do know it's going to get warmer. I don't know how much our winter freezing days are going to diminish, but I know which species are going to be the first ones to suffer from that by having this data. So, yeah. Yeah. Thanks, everyone.
Down is forward. All right. Hi, everyone. I am Hannah Demler. I'm a restoration technician with the Nature Conservancy in Wyoming. Does that sound okay to everyone? Okay. And I'll be presenting our work on understanding seed dormancy and availability to improve forb establishment in Wyoming. Working with me on this is the conservation scientist in Wyoming, Maggie Eshelman, and Olga Kildashelva, who's our innovative restoration program manager. And I would like to thank the Wyoming Department of Environmental Quality, who has provided most of the funding for us to do this work. So I'd like to start by acknowledging that Indigenous peoples and nations, including Eastern Shoshone, Crow, Cheyenne, Arapaho, Seven Council Fires, and Shoshone-Bannock peoples live throughout and around the land that we now call Wyoming. We recognize that they have lived with and stewarded these lands since time immemorial and acknowledge that the lands we work on are the stolen ancestral homelands of countless Indigenous people who were killed, removed, or relocated. And we would like to voice respect to the enduring relationship that exists between these peoples and nations and this land. So a bit about where we work. The classic plant community in Wyoming is the sagebrush steppe. A lot of Wyoming actually has really healthy, intact sagebrush steppe ecosystems, but they are threatened by things like natural resource extraction and energy development, so there is a growing need for restoration in Wyoming to keep the land healthy and connected. Despite from afar, some of these ecosystems can look a little bland, but they're actually teeming with diversity. Importantly, native forbs are an understated yet vital part of functioning ecosystems across Wyoming. Everyone here probably knows why forbs are important, but a couple examples: they attract a diversity of pollinators, and they're also a necessary component of healthy wildlife habitat. A really meaningful example in Wyoming is that they are essential in the diet of greater sage-grouse hens and chicks. And for those of you that don't know, greater sage-grouse are emblematic of the sagebrush ecosystem, and they have become sort of a symbol for conserving sagebrush steppe across the West. And so there are quite a few limitations to successfully establishing forbs on restoration sites in Wyoming. There's a lot of great work being done, like in the Great Basin, on forb establishment on restoration sites, but Wyoming does sit in a unique ecoregion and also hosts a unique set of forbs that don't necessarily overlap with some in other arid lands in the West. So unfortunately, despite their importance, forb establishment from seed is often really limited on restoration projects in Wyoming. This stems from several factors, including there's just not native seed available, they're really difficult to collect in large numbers, and they're also difficult to grow. A lot of them have slow and irregular growth patterns, and species-specific seed dormancy characteristics that can make them hard to grow when and where you want them to. So our objectives for this work are to understand how to germinate and grow native forb species to Wyoming that are currently unavailable, and we hope that this will inform future seed production and support the use of these different forbs on restoration projects in Wyoming. And I have one slide at the end where I'll talk about our objective to understand seed availability and demand within Wyoming. So one of the biggest biological barriers to getting native forb seeds to grow is seed dormancy. And as everyone here probably knows, seed dormancy is an evolutionary adaptation that prevents seeds from germinating during conditions that are not suitable for plant establishment and growth. So in natural settings, this is a good thing, but for restoration practitioners and seed growers, it can be a challenge. So many native forbs to the sagebrush steppe have developed specific seed dormancy mechanisms. Common ones include physiological dormancy, where the seeds need cold stratification or a chilling period in order to germinate, or physical dormancy, where the seed coat needs to be scratched or abraded in order for seeds to take up water and germinate. There are other more complicated types of dormancy, like combinational dormancy, where the seeds can have both physiological and physically dormant aspects, and also morphological dormancy. And so seed growers and restoration practitioners have to be able to manage seed dormancy in order to get these important species to germinate and grow when and where we want them to. So first we had to choose some target species to just begin this work, and we had some priorities for choosing target species. First, we wanted to work with species that have unknown seed dormancy classifications. We also wanted to work with species, obviously, that we thought had high potential to do well on restoration sites, that could grow well on disturbed areas across the state, and also species that were important to greater sage-grouse habitat. So we did a bunch of research. We consulted with partners at local and state BLM offices and went through relevant literature to get this sort of master list of species that we think would be good targets for Wyoming restoration. And then we researched seed availability, dormancy and germination characteristics, and any germination information we could find on these species or closely related congeners to sort of get an idea of where to start. And then we ended up choosing species that aligned with our priorities and that we could obtain seed for either initially through purchase or later through wild collection. So we have completed seed dormancy experiments on 2 native species, the first being Astragalus convolvulus and the second Macaranthera grandelioides. We've classified the previously unknown seed dormancy and germination requirements for these species. We used the Baskin and Baskin methods for seed dormancy classification, where we systematically assess different seed traits in order to determine the germination requirements. So in the next couple slides, I'll just go through the highlights of the experiments we did on these 2 species. So the first species that we investigated was lesser rushy milkfetch, or Astragalus convolvulus. And we started with an imbibition trial. So imbibition is seed uptake of water. And this figure on the right here shows that we had a set of seeds that we scarified with sandpaper and a set of seeds that we just left bare unscarified. And we monitored percent mass increase, which is how we're monitoring water uptake of these species. And we found that after 48 hours of imbibition on moistened filter paper, the scarified seeds took up significantly more water than the unscarified seeds. We followed this up with a germination trial, and scarified seeds germinated really well, greater than 75%. So that indicates that this species is physically dormant and has a hard seed coat, which is not that surprising because it's an Astragalus. So scarifying seeds by hand with sandpaper is pretty low throughput, and so we wanted to test some different mechanical scarification treatments. We used this Forsberg scarifier, and we had really low availability of the convolvulus seed, so we did some initial tests with a different Astragalus species, Astragalus cicer. So this machine here has a drum that's lined with sandpaper that you can put your seeds in, and then it goes inside that other cylinder that has rotating paddles that spin the seeds around against the sandpaper. So we tested a bunch of different levels of abrasion and durations of abrasion on this figure here along the x-axis. You can see those. And then we did a germination trial, and we measured cumulative percent germination of seeds that had gone through all these different treatments. And we saw across the board, regardless of level or duration of abrasion, really great germination from all these different treatments. So we did use that as sort of a starting point. This figure is showing Astragalus cicer is the green and Astragalus convolvulus is the brownish color. We only could do really small volumes, so it didn't work great in this machine. But we did test some different treatments, and we saw not quite as good a germination, but the convolvulus did perform pretty similarly to the cicer, so we think that with more seed, we could optimize mechanical scarification for this species. So the next species that we investigated was Rayless tansiaster, or Macaranthera grandelioides. Again, we started with an imbibition trial and found that untreated seeds imbibed water just fine, but in a following germination trial, we did not get good germination of any seeds that were untreated. So we expected this species to have physiological dormancy. We conducted a cold stratification experiment where seeds were incubated at 3 degrees Celsius in a refrigerator for durations of 1, 2, 3, and 4 months. And we also included a gibberellic acid treatment, which is a plant hormone that can induce dormancy break in some physiologically dormant species. So the good news, all of our cold stratification treatments did lead to significantly higher germination than the control. In our 2, 3, and 4 month treatments, germination was over 75% in the subsequent germination trial, but we did find that our gibberellic acid treatment did not significantly increase germination over the control, indicating that at the concentration that we used, which was 1 millimolar in this experiment, it was not a viable means of chemically inducing dormancy break for the Macranthera grandiloides. And we have published the seed dormancy characteristics and germination information that we found on these 2 species on the Native Plant Network Propagation Protocol database. And we hope to get that information out in some other ways to seed growers and users, just to have that available for anyone who's interested. So we don't really know if these species are going to be good targets for restoration, even if we can break dormancy and get good germination, unless we can get them to establish well in the field. So we do have some small field plots set up at one of our mine reclamation sites, some pictures of it here. And last fall, we sowed bare Macranthera seed and scarified astragalus seed. And if the snow ever melts this spring, we will go out and monitor for emergence and germination, and then throughout the summer we'll monitor growth of those plants. We are continuing this work. We do have some ongoing seed dormancy experiments with 6 more species, Astragalus pershii, Erigeron cespitosus, Ipomopsis congesta and spicata, Fuchsia americana, and Penstemon arenicola. And we also expect that each of these species will also demonstrate either physiological, physical, or perhaps combinational dormancy. So we're going to be doing similar experiments as we did to the first 2 species on all of these as well. Next steps, we want to continue sharing and publishing our species-specific seed dormancy and germination information. We also want to keep testing scalable dormancy break treatments, as well as field establishment for our target species. We are working on selecting more high-priority species to work on in the future, and collecting seeds for more seed dormancy experiments. And ultimately, we hope to leverage this work to get more of these species into commercial production and grow interest in them for use on restoration projects in Wyoming. And so now I just want to touch briefly on our sort of larger scale work to improve native seed supply in Wyoming. And there was a talk yesterday on the Wyoming native seed strategy, so you got a lot more information about this if you attended that talk yesterday. But we kicked off this work by my colleague Maggie Eshelman last spring sent out the Wyoming Native Seed Survey with the goal of reaching out to seed users and producers across the state to better understand native seed availability, demand, and associated policies. And some highlights that we gleaned from those results are that at least $3.9 million is spent on seed for revegetation every year in Wyoming. 73% of the respondents did say that their organization does have a policy for the use of native over non-native seed. However, they also said that the lack of availability and the cost of native seed were the greatest barriers to creating an ideal seed mix. Additionally, almost 50% of the species that were listed by respondents as desired species that were unavailable were forbs, so that supports our work to continue figuring out how to grow native forbs. And also, many restoration projects in Wyoming are being planned in advance for things like oil and gas, mining reclamation, construction engineering projects, as opposed to in response to natural disasters like wildfire. So that does lower the volatility of seed demand in Wyoming. So in the future, we hope to facilitate things like improved seed planning, feedback between seed growers and seed users, as well as communication with practitioners and land managers just about native plants. And yeah, all of this work together, we hope, is progress towards just better and more native seed being available in Wyoming and ultimately better restoration outcomes. And that is all I have. And the seed survey report is available there for anyone who wants to read the full thing. I should have brought physical copies, but I forgot. So.
If you find out one has like combinational dormancy, maybe needs to be stratified and scarified, is there like a standard order to do them in, or does it depend on species, or?
So we would probably start with the scarification, just because we do the imbibition test first, and then we would, follow up with cold stratification. But it might depend on the species whether one would be better before the other for like actual, if you're treating the seeds to put out on restoration projects. But yeah.
Since you're sort of picking the seeds in the restoration project to germinate when maybe it's not perfect environmental conditions sometimes, have you noticed any in your monitoring, like things maybe dropping off because maybe they germinated at the wrong time?
So we actually don't have any field data yet. We're just at the very start of this, but yeah, that is a concern, is that things are all germinating at once, and then they realize that it's not great to be growing right then. That is a concern. But a lot of people in Wyoming will just sort of write off species if they don't see them like within the first year or two. So at least like getting something out there for the practitioners to look at and be like, okay, this species is at least growing. Yeah, just like getting that interest level higher is good.
In the trials, did you have some scarified and some not scarified seeds, or did you scarify all of the?
In the field trials, we scarified all of it. One, we had pretty low number of seeds, so we didn't really want to put out unscarified seeds that we didn't really think were going to do anything. But ideally, yes, we would have also put out unscarified seeds to see what they did.
Do you have any guidance on how you select your scarification method? Acid versus boiling water versus.
We just started with sandpaper because it's easy to do in the lab, but we have not tried acid scarification. That is something that we were looking at, but again, we didn't have a ton of seed for these first couple experiments, so we just started with sandpaper. But it would be really interesting to try some different methods. Also, the scalability too. We wanted to do treatments that could be carried out at a large scale, so that if someone did want to use these species, like for seed production or for putting out on restoration sites, you could do a bunch of seed at once and have it ready to go.
Cold stratification scale up. How would you do cold stratification scale?
So you can do big mesh bags of seeds with buckets of water and refrigerators. We have not tested any large-scale scalable cold stratification treatments, but I think that agriculture, and especially some cut flower production stuff, does do stuff like that. So I think that we could get information from people who have methods for large-scale cold stratification.
Next up is Rae Robinson.
Awesome. Well, yes, my name is Rae Robinson, and I work as a lab manager in the Wetland Ecology and Restoration Lab at Utah State University, and I'm really excited to talk to you guys today about some of my research looking at dormancy break of widespread native wetland plant species. So we've already heard a little bit about dormancy today, but we know that dormancy prevents germination in unfavorable conditions. It's a way to minimize spatial and temporal risk, which in wetland environments, this might be particularly important as conditions can be very dynamic. As you can see, wetlands can be very dry, but they can also be very inundated. Species that exhibit dormancy might allow for their dispersal, and that dormancy would prevent germination and then allow them to disperse either by water, so this is alkali bulrush there on the screen, or by birds spreading hundreds of miles. So although it's advantageous, dormancy can make seed-based restoration in wetlands very challenging. Now, seed-based restoration is increasingly being practiced and often needed in wetlands. Wetlands are very invasion prone, so if you can imagine, wetlands often exist at the bottom of a watershed, so you're seeing high influx of nutrients, pollution, and then also invasive plant propagules. And this is just a picture from a wetland that I work in, Great Salt Lake wetlands, which has been invaded by Phragmites australis. Phragmites is a problem not just in Utah wetlands, but also in wetlands across North America. And you can see here, there was actually a treatment to remove Phragmites with mowing and also herbicide, but this green grass coming back, you can probably guess, actually is Phragmites. So in these situations, native plant communities are slow to recover, or they might not even recover. And for that reason, active revegetation, usually using seeds as a scalable strategy, is needed. As we know, outcomes of these seeding efforts, though, depend on the ability of native plants to quickly germinate and emerge. And therefore, we need effective dormancy break of these native wetland plant species for both fast and high germination, but also germination under broad conditions. And I'll point out from a practitioner standpoint, successful dormancy break is the efficient use of restoration resources, which we know can be very limited. So there's been some great research. Julie already kind of filled us in on the power of seed and seedling traits. And there's been some of this work in wetlands, although I feel like wetland research on seed and seedling traits kind of falls behind some of our upland counterparts. But I want to highlight some of the research of Emily Tarsa, who is a professor now at College of Coastal Georgia. She was a former PhD student in our lab. And she looked at both intra and interspecific variation in seed and seedling traits of 5 native wetland plant species. So there's definitely a foundation for this research, but we lack information for the wide array of wetland plant species. So our research objective is to start to build portfolios for each of these species to improve our basic knowledge and also to inform revegetation practice. So here are some of our plant species that we are investigating in this research. We have 34 species total, representing 12 different families. We collected a lot of these species from Great Salt Lake and Utah Lake wetlands. These are emergent wetlands, meadow communities, playa communities, and then also some of these drier wetland to upland transition areas. We asked, to what extent do these species exhibit physiological dormancy, and what strategies are needed to break physiological dormancy? So we conducted a series of 3 experiments. The first experiment, we looked at how a 4.5-month cold stratification treatment affected germination compared to a room temperature control treatment. And there was nothing too special about the 4.5-month treatment. These seeds were already in cold stratification, so it was just a good opportunity. And the way we do cold stratification, we put our seeds in these little mesh packets there, bury them in moist sand and moss layers at 2 degrees Celsius. And then at the start of our germination trial, we basically grid them onto saturated sand. A lot of these species need high light, so we're not burying them or anything, under conditions which were 32, 15 degrees Celsius and a 14-hour photo period. So after we did this first experiment, we found that some of our species were minimally to non-dormant, some of them were dormant. So then we conducted another experiment where we looked at how a 0, 15, 30, and 45-day cold stratification treatment affected germination. And then also for a subset of our species, species in the sedge or Cyperaceae family, we treated them with a 0, 24, or 48-hour 3% bleach scarification treatment, followed by a 0, 30, or 90-day cold stratification treatment, and again, measured that germination response. I'm going to talk you through some results and just highlight some main trends. I do realize it's getting a little bit later in the day, so right here we have on the x-axis the day of the experiment. So this is when we are going into the lab, counting and removing germinated seeds to get that cumulative germination. On the y-axis, the cumulative germination is that cumulative germinated number divided by the bulk seeds in the tray. And then viability is represented by that dashed line. So these are raw data that I'm showing you here. And the blue color is the cold stratification, yellow represents the control. You can see for our Castanea species, you get very fast and high germination with the cold stratification treatment. Very similar trend with our verbena species. For Symphyiotrichum ciliatum, we see that too. The cold stratification treatment really increases germination. And then you see actually in the control maybe a slightly different trend where ultimately by the day 30, the same max germination. However, the rate in which it got there is very different. Now, some of our species actually are minimally to non-dormant. So if you look at Euthymia occidentalis, for example, those lines track pretty closely. Same with Rumex bertimus, maybe you're getting a little bit faster germination if you treat it with cold stratification. And for Phragmites australis, it was actually something kind of surprising. So if you remember, Phragmites is not a native species. We simply use this as a point of comparison in these experiments, where actually the control had higher germination. I guess in hindsight, it's maybe not that surprising. So this species produces tons and tons of seed. Seeds are very small, very thin seed coats. So perhaps in the cold stratification treatment, those seeds were actually using up their reserves, and then they didn't just readily germinate at the start of the trial. Overall, maybe this is reflected of other people's presentations, very species-specific results. All right, now for experiment 2. So the dark blue color represents the 45-day cold stratification treatment. The royal blue is the 30-day. The green blue is the 15-day, and then the yellow is the control again. So for Biden-Cerneux, kind of get a clean trend here, where if you increase the number of days in cold strat, you get that higher germination, followed then by the 30, the 15, the 0. But this, of course, doesn't play out in all of our species, even within the same family. So Helianthus natalii, the 45-day performs higher than all of the other treatments. Helianthus annuus, 15 days or longer equals better germination than that 0-day untreated control. I'd like to share the example of the Juncus gerardii, where if you just simply look at day 5, just like stark difference between the germination. So very high for that longer cold stratification time of 45 days, very low for the 0. And then compared to Juncus torii, just seeing how different these germination trends are. So we are seeing different trends even within a genus. All right, lastly, as for our experiment 3, so if you'll remember, we had a 48-hour, 24-hour, and a 0-hour, which was actually just a 24-hour water soak for our scarification treatment, followed by cold stratification of 90, 30, or 0 days. So you can see we really hit the sweet spot here with the alkali bulrush. So that is 24 hours followed by 24-hour bleach followed by that longer cold stratification time, so that 90 days. And yeah, this was pretty exciting for me because this is a species that a lot of the wetland managers that we work with like to sow this species. So, and look at that just even compared to the control. It's a really stark difference. Chenoplectus acutus, some combination of bleach treatment with at least some cold stratification treatment improves germination. That's much higher than that control treatment. So that's just no cold stratification days. That yellow color is much lower. So for our Carex species, so these species are in the sedge family. They're not bulrushes like the species on the first row. They're true sedges. It was a little bit of a different story. So you can see here for Carex nabrasensis, no bleach was required, and even not putting the species in cold strat seemed to have high germination. So this was kind of interesting, and this was different than some of the literature I had read about this species. And then you can see about intermediate germination for the 24-hour bleach for this species, so it's almost too rough. And then the 48, you can see, is basically around 0. So my thought about this is that these scarification treatments were too rough for some of these sedge species. And then we probably kind of hit a sweet spot again with this Carex prograsslus, where the 24-hour was kind of the highest compared to the other treatments. So overall, species specific, some species need both, some species might not need any. So, and sometimes adding bleach is definitely not helpful. Just to summarize what we found from these experiments. Some of these species seem to be minimally to non-dormant, so this is good news for practitioners who might not want to put in that preparation time to prepare them to be sown in the field. So here's just an example of some of those species, but we definitely need to consider functional trade-offs. So species that readily germinate and maybe are not deeply dormant might have trade-offs in terms of they might establish cover quickly, but then they might senesce or they might die off. So at least from working in my system where invasion resistance is really important, we definitely need to consider these functional trade-offs. Some species have low to moderate dormancy, so they'll require some length of cold stratification. This was about 75% of those 19 species we tested in our second experiment. And some species require longer cold stratification, scarification, or both, and these species might tend to have deep dormancy. And then this point has already been made, but perhaps these species that have deep dormancy, maybe this is a good thing in that they'll provide resilience in future years at the restoration site. Next steps for me, I'm going to look at time to 50% germination, maximum germination, and dormancy index in my data analysis. We're also working on a seed collection and cleaning dormancy breaking guide for wetland species in the Intermountain West, which will be released through our USU, Utah State University extension. So be on the lookout for that. So in conclusion, breaking dormancy should definitely be used with other best practices for seed-based restoration, and I want to highlight an example of this from last field season. So last field season, we were able to work with our state agency partners. We revegetated several acres in Great Salt Lake wetlands, and we were sowing alkali bulrush and saltgrass mostly. So that was in June, and then in September, this is what some of our monitoring points look like. So on the far left is an unseeded control, which you'd see is pretty bare. And on the right is the distant seeded area. So this was actually a huge win in my eyes. It was really great to see this. This green lawn grass here is actually the native saltgrass that we sowed. So this was really awesome to see. And we attributed this really incredible success to the 9-month cold stratification treatment that the managers implemented to break that dormancy. We also used a farm disc ahead of our marsh master that was seeding. So what we were seeding had already been disked, and that was to create microsites. And we definitely saw seedlings germinate and emerge in both the low spots and the high spots of those microsites. So that was pretty cool to see. But before we did this, there was a multi-year consistent Phragmites management, which really just gave the native species a chance. We used a very high seeding rate, between 40 to 230 pounds per acre, which I think definitely helped. And we were also saved by August and September rains, which really provided some really needed soil moisture. We hope to continue to build our knowledge of the seed-to-seed transition, especially for these wetland plant species. Looking at light, salinity, I could probably throw in temperature and also water potential in there, and also seeing how these different seed and seedling traits play into all of this as well. Just want to thank all the people that support this research and take anyone's questions. Yeah.
I was wondering where you actually source your seed from.
Yeah, so we collect a lot of our seed ourselves, just students and technicians in our lab from Great Salt Lake and Utah Lake wetlands. Yeah, which is kind of a fun process. I get to go out and scout the populations and track the phenology, and then eventually collect, and then involved in the cleaning process as well. I will say we also partner with, occasionally our state agencies will give us seed sources from their warehouse, and we also work with a local collector who will help as well.
Were all those species being drilled, or were some of them just being broadcast on the surface?
Yeah, so they're being broadcast. Yeah, with.
I'm trying to think about like the dormancy requirements and like sometimes seeding in the wetlands, and then does the seed actually kind of stay put, or does it, move around in the water and that kind of thing?
Yeah, so I was in a couple years ago, I was involved in an effort where we actually hydro-seeded. So there's like a tackifier with a botanical glue and did all of that. And that was supposed to keep the seeds in place. I think recently it seems like that the consensus is like maybe that doesn't seem to work in wetlands. Like you go out a few days later and there's like no evidence of the tackifier keeping the seeds in place. So for that reason, at least for this effort, our collaborators just decided that we were gonna try the disking and just broadcast seed after that. Yeah.
Why did you settle on 3% pH, not something higher?
There's been some previous research where we got the 3% from, so figured that was a good starting place. Yeah. Yeah, thank you.
Push down to go forward.
Okay. Sorry. It's good to be with you. My name's Kyle Cook. I work at BYU. Well, I'm a master's student at BYU. I like to pretend that I work. But I work in Matt Madsen's lab. I don't know if you're familiar with him, he does a lot of work with seed coatings. And today I want to talk with you specifically about using seed coatings to improve restoration success of winterfat. So winterfat, Crescentinacovia lanata, this is a valued rangeland plant, mainly because of its high protein content during winter months. A lot of plants lose their crude protein or it's inaccessible to animals during this time. This plant maintains high levels of crude protein during those months, so it's really helpful to get those animals through the winter. Like many other native species, however, this has been historically overgrazed, especially during the homesteading era, and areas that were once dominated by winterfat have been, in those areas, winterfat has been reduced. It still remains dominant in many salt shrubland ecosystems, but it's been reduced in some areas, and unfortunately it has seen limited recovery. There are a lot of barriers to winterfat restoration, and we'll get into a lot of those here in a minute. But one of the bad things about this is niches that were once occupied by winterfat are now being occupied by this lovely plant right here, halogeton. Halogeton occupies very similar areas as winterfat, but it's not nutritious. It's toxic to many animals and to livestock. So we see livestock losses from this plant. Additionally, it can change the microbial community in the soil and make it less conducive to winterfat establishment and more conducive to further halogeton establishment. So it's a big problem. There's a need to restore winterfat for sure. So here are some of the barriers to restoration. One of the big ones is just seed flowability. So if you look on the bottom left, that's a close-up picture of the fruit of winterfat. It's a utricle that's covered with a dense covering of those fine silky hairs. And what that means is that you try to run these fruits through mechanized equipment, mixing equipment, and you get this bridging effect like you see in that really fancy beaker setup that I set up in the lab. But they'll bridge and they'll clog equipment, and as a result, a lot of managers just see this as a really big inconvenience, see it as being impractical rightfully, and they exclude it from their seed mixes, or it's included at really low seeding rates and rates that are really too low for stand establishment. So that's one big problem. Another problem is even when we are able to plant it, we often end up with what you see on the right, just tiny little seedlings that germinate and try to emerge at the wrong time. And they experience high mortality due to a lot of factors. Freezing, pathogen attack, winter drought, Specifically, pathogen attack is an issue. If you look again at the picture on the bottom left, those hairs provide really, really high surface area. And that surface area, that high surface area is prime habitat for pathogens. So they're really subject to that. So we can't plant it often. When we do, it doesn't work very well. Another limiting factor here worth noting, even though our research didn't focus on this, is just short shelf life. Try to store it and within a couple years, most of it's dead. So that's definitely a point for future research. Quantifying when viability goes down and then also just keeping that up. So this is important to note with flowability since it's such a big issue. What have we tried? We've tried a lot of things and some of them work really well, some of them don't. They all come with their trade-offs. So one of the main things we've tried is just mechanical cleaning, removing the seed from the fruit through a variety of methods. And one of the good things about this is you can remove the seed from the fruit and then no flowability issues, right? You can put it in the seed mix just like anything else. But a bad thing about doing that is there's literature to suggest that doing so damages the root cap of the seed, and then we have viability and or vigor issues of seedlings that establish. So, like I said, pros and cons with that one. Another method is using carriers like rice hulls or cracked corn. Mix those in just like this is used with other fluffy seeded species. Our experience with this, we tried it in the lab with just a chest broadcaster, and we had to put a ratio of 5:1 carrier to winterfat seed to get it to flow. So a lot of carriers for a little seed. And then also some of them can attract rodents and promote herbivory. So there's problems with that. This one is really cool. So flash flaming is another method that's been used to improve flowability for this species. Essentially what that means is we'll take these fruits and we'll expose them briefly to an open flame, which will singe down the hairs, create a more uniform product, kind of like what you see in the middle picture there. And then that more uniform product, it has slightly improved flowability, still not great, but you can apply a coating over top of that and it'll flow through anything. So that's a really cool method that the grad student previous to me did a lot of work on. One of the issues with this is that drum right there with the open flame is 31 centimeters, and it's just, it's not scalable currently for this species. So you cannot flame very much at one time. It's time consuming, it's costly, just not scalable. So what we're proposing is using seed coatings as a solution. The ability to apply coatings to winterfat without a pre-treatment, like flash flaming or like threshing, that's new. In the past, people have tried to coat winterfat just like this, and it's just a clumpy, muddy mess. Those hairs just don't grab onto coating material very well, or they stick together, and you end up with a winterfat seed that's that big, you know. But through about 2 years ago, we tried it. We just went in the coater, pointed due south, kicked the thing 3 times, and tried all sorts of different methods, and we were able to find a way to put coating material on the fruits with no pre-treatment. So that's what we're doing now. And this improves flowability. The coated fruits do not have issues with bridging. They'll flow just as well as anything else. And the coating that you're seeing is our blank coating, meaning it has no active ingredient like fungicides or any other chemical. It's just calcium carbonate, which is an inert, low-cost material. We got a ton of it for, literally a ton of it for 75 bucks, so very cheap. And just doing this, applying an inert coating, can increase planting opportunities. But then you can further improve it by adding active ingredients, targeted coatings to the blank coating. So the issue we talked about earlier is that when you plant winterfat in the fall, this species will germinate really quickly when conditions are right. So you'll have a warm spell in late fall, winter, any time during the winter, and these seeds will think it's time to go, they'll germinate, and then they will die with the oncoming pathogens and freezing that winter poses. So what we do is we put a hydrophobic coating on the outside of this seed, which is a watertight seal, so that you can plant that seed in the fall. That hydrophobic coating will keep the water away, prevent imbibition until the spring when that coating has degraded. Then when conditions are more right, that seed can germinate and face a nice easy spring instead of a tough winter. So we have one coating that can solve 2 problems, flowability and poor seedling establishment. So here's what we did. I'll kind of pick up the pace through this stuff because, you know, we like the other stuff better. But our first question was, do coatings inhibit emergence? By adding a barrier around the seed, are we going to trap that embryo within the seed? We want to make sure we don't do that. Do coatings change planting protocols? By adding an extra barrier to the seed, are we imposing extra energy demands so that if we plant it deeper, it's not going to be able to emerge? So to answer these questions, we did seeding depth trials in the lab and the field, just looking at the blank coating with no active ingredient. Then we wanted to see, how do these seeds perform under realistic conditions? So we were able to participate in some habitat improvement and mine reclamation projects to just test it out in its current incomplete form. And then finally, can hydrophobic coatings really improve emergence? It's a nice story, but let's put it to the test. So we did hydrophobic trials. So this is our coater right here. This thing is awesome. I've spent way too much time with this over the last couple years. And just a brief description of how it works. So this is a 31 centimeter drum. You put the seeds down into it, and the seeds rotate on that big disc on the right. Your liquid material is applied to that small disc, which is the atomizing disc. That disperses your liquid material evenly across the seeds. And liquid material is generally a binder. It's a glue that will hold the powdered material onto the seed. And then you just apply whatever powdered material you want to coat straight onto the seeds as they're spinning. So here's our first study. This was our seeding depth trial in the lab. So we compared uncoated fruits with blank coated fruits at 4 planting depths, basically from soil or surface sown to about a ½ inch deep. Within loam, and sandy soils, we had 8 replicate blocks of this. We used hand furrowers that imitate a seed drill. They make a really precise furrow, very small furrow at a precise depth, and then we pinched it closed. The growth chamber was set to 15 degrees Celsius on a 12-hour light and dark cycle. All flats were watered equally, and we counted emergence weekly for 3 weeks. It's important to note that nothing came up after 2 weeks, so it is a really quick germination and emergence process. And then for the field trials, we just tried to replicate this across 4 field sites. We were up at Rio Tinto on a tailings impoundment. We were at Santaquin, which is a wildlife management area, and then Sage Valley and Enterprise, which are both post-burn restoration sites. And these were the same sites that were used in our hydrophobic trials we'll talk about a little later. We used tractor-pulled furrowers to install those furrows, and then within the furrows, we used those hand furrowers, same as in the lab trial. That was to ensure that planting depth was consistent in the lab and the field. We planted in the fall of 2021 and counted emergence in the spring of 2022. And here's what we found. So in the lab trials, on your y-axis, you're going to see soil type and % emergence. And on the x-axis, you're going to see our planting depths. Light gray is control, non-coated. Dark gray is coated. And we saw in part what we expected, which is that shallow planting depths are going to yield the best results for both treatments. But one thing that was a surprise was on the soil surface, the coatings seem to do a lot better than the non-coated fruits. And we guess that this is because those coatings allow the fruit to settle deeper into the soil. If you put a winterfat fruit with lots of fluff on it on the soil surface, that fuzz kind of suspends it up above the soil surface. So it's not going to have as much seed soil contact, not going to have as much available water. So kind of a cool finding there that we didn't anticipate. And our field trials showed pretty much the same pattern, so definitely worth looking into. So then our next step was we wanted to take those blank coatings and try them in real world scenarios. So we had five sites that we planted at, again at the Rio Tinto tailings impoundment, mine reclamation site, and then 4 habitat improvement projects. They were diverse. There was big game improvement, there was prairie dog translocation, there was sage grouse, but lots of mechanical treatments being used there. So we had those sites, and we had 1 acre plots set up at each site. Half was control, half was coated. And we broadcasted seeds at 7 pounds PLS per acre. Control by hand, because that's kind of all we got for broadcasting with control seeds right now. And then coated with just a chest broadcaster. We planted in the fall of 2021, and we counted in the spring and fall of 2022. One other note here is we planted just before the mechanical treatments were applied, like days before. And that was consistent across all 4 habitat sites. The tailing site, there was no mechanical treatment. And here's what we found. So as far as density, on the y-axis you'll see density, on the x-axis you'll see our treatments. There was no difference. So whatever cool magic effect we saw in the depth trial with those surface zone seeds, blank coating was way up here, somewhere in the real world that got lost. Which is okay, because this is similar, and we can plant coated seeds and we can't plant non-coated seeds. So it's still a good finding. And then with, we did see a difference in frequency. In other words, on a control plot, we saw patches of seedlings like this. They're adapted to be windblown, so they were windblown into catchment areas against stumps, and they grew in high numbers in those sites. And then the coating kept the seeds in place. You had more even stands. So that's just an interesting note to keep in mind. And then finally, I'll touch on this really briefly. On the right, you can see what I'm talking about with those catchment areas. We saw all of our control seeds were in places like that. They did not grow spread out. It was in catchment areas. So those are important for untreated seeds. And then a few of the habitat plots did not get their mechanical treatment. For one reason or another, they couldn't take equipment out there. And in those sites, we did not see very much emergence. So in the middle picture, you'll see an excavator track, and then the seedlings would grow right up there where the soil disturbance took place. So we know soil disturbance it has its good things and its bad things, right? But in this context, we did see that that helped promote emergence in both treatments, control and coated. All right, finally, this one luckily is a simple story to tell. So we tried our hydrophobic coating again to delay germination until spring when conditions are good for seedling establishment. The 4 sites, same as in the depth trial. We used control seeds, blank coated, and hydrophobic coated seeds. Same tractor pulled furrowers and the hand furrowers. We planted them all at about 1/8 of an inch deep, so consistent planting depth here. And our hydrophobic material that we applied was a 7% ethocell in acetone solution. And here's what we found. So you'll see total emergence on the y-axis and then treatment on the x-axis. Similar between the control and the blank, but the hydrophobic does seem to be doing something, right? Now, there are a couple things to note here. First, we did not measure the timing of germination. So in theory, we're saying this hydrophobic coating is delaying germination, and that's what's improving emergence. But we can't say that for sure since we didn't test the timing of germination. What we can say is that we applied a hydrophobic coating and emergence was better, so research into hydrophobic coatings is definitely warranted. Another thing to note is that we are working from the ground up here, so if you look at the plants per meter, we are still really low. This equates to about a 2.4% emergence rate for the hydrophobic coating, and somewhere around 0.8 for the control. So we have a long ways to go still, but this is, it definitely shows merit for future research. So just in case you didn't know which one we were talking about there. I think, I'm gonna speed through this because I think I'm running out of time here. In summary, what we know is that coatings don't inhibit emergence, so we can improve planting seed handling. We can plant these seeds easier and not worry about trapping that seed in there. And we can use hydrophobic coatings potentially to delay germination of fall sown seeds until spring and hopefully produce better plant establishment that way. And with that, I will take any questions that you have. Yes?
It was interesting to me to see like the sort of the placement of the plants based on they had landed where you put them when they were coated, but then they kind of went up against those like logs or whatever when they weren't coated. I was wondering if you thought there was any advantages to the uncoated kind of like, I can almost see like a snow fence kind of thing that occurs when they all are growing in that nice line up against old logs or whatever.
Oh, definitely. I think there's an advantage to that patchy growth pattern. Having a stand that has a lot of seedlings where conditions are right, there are advantages to that. I think the real important implication there is if you're going to go and plant winterfat that's not coated, you want to make sure that that site you want to have winterfat on has something like a snow fence, or it has catchment areas for that seed to blow into, because it's going to go. I mean, it's adapted for that.
Yeah.
Good point. Yes.
I was just wondering, so for the soil conditions of this type of plant, does it prefer like calcium-rich soils, I guess?
Calcium rich, not necessarily to my knowledge. It's tolerant of alkaline soils. You know, that's one of the things that goes for it. But as far as, I don't know of any advantage to calcium rich soils. I think, are you kind of referring to the calcium carbonate coating being part of?
Yeah, I was just wondering if that skewed the results at all.
Yeah, we haven't looked into that specifically. Calcium carbonate was an awesome thing because it's cheap. If Captain Crunch powder was cheap, we'd use that too. Just anything to get it out onto the landscape, you know. Good question though, thank you. Yes.
Are you guys seeing better results delaying germination with the hydrophobic coating versus something like abscisic acid? Is that?
That's an excellent question. So again, the grad student before me started looking at delaying germination using abscisic acid, soaking it in abscisic acid, and he didn't see anything with that. So there was no difference. That's not to say it couldn't work, but that's what his findings were. In addition, the cost is way higher with the abscisic acid, so we pursued this route, and so far so good.
Yeah.
Yes?
To your knowledge, has there been any plantings of winterfat using a native seed drill?
Yes. I run into them all the time, actually. What I run into all the time, though, is other species, and then like there's one winterfat seed at very low rates. So it's almost like, why even plant it at those low rates? You know, it's going to have to compete with all the other species in that mix. And again, the reason for that is just because if they plant too much of it, it's going to gum up the works of that piece of equipment, going to clog up and just slow down the whole project.
Have y'all
Yes.
looked into using graphite to kind of lubricate some of those seeds so it doesn't gum up? We use it with all of our fluffy seeds. We mix it into the seed box, and then we plant it.
That's really interesting. I have not used graphite and you would apply that to the fruits and then?
So we'll put the seed drill, there's a slick seeded box and a fluffy box.
Yes.
So in the fluffy box, we just sprinkle in graphite. It's a powdered graphite. Mix it in. It helps lubricate and cut down on the static electricity.
Awesome. No, I haven't done that. You're talking next, right?
No.
Yes.
You did say your emergence numbers are horribly low. Have you got an inkling why the emergences are happening, and are you checking whether it's the germination failing below ground or the plant actually failing to then emerge from the soil surface?
I don't want to always pull this card, but we did have a really dry year across those sites. You know, germination of this lot was 54%, and they had been gathered the year before, and again they have a really short shelf life, so it's quite possible that we lost a lot of viability on the shelf. The drought probably played into it. That's at this point, that's my best guess. Yes.
Were you able to determine like the vigor of the plants that did germinate from your coated seeds?
So with the habitat plots, we did go back and measure survival through that first growing season. We didn't look at vigor specifically, but it was actually really, really good. And I wish I had those numbers, wish I had prepared that too, but it was higher than we expected. Quite a few of those plants survived through that first growing season. But the other studies we did not measure. We didn't take that second measurement.
Anybody else? Well, thank you.
Thank you.
And we do have a fifth speaker, and Katie VinZant from the National Park Service. There you go. And push down.
Want to sleep yet? Because this presentation will put you there. No, let's hope not. Okay, so my name's Katie. I work for the Park Service, but I did this work back when I used to work for the Forest Service. But the brains of this operation really is Susan Frankel, who works for the Forest Service in the Pacific Southwest research station. This case study is going to take place on the Angeles National Forest, which is down in LA, probably the most urban forest ever created. This project occurred on a right-of-way, several right-of-ways for large-scale transmission lines that were coming across the forest and also pipeline right-of-ways. They like to joke on the Angeles that we don't grow trees, we grow transmission lines. It's very urban forest. So we got really jazzed. We had seen some really great success with container plants in our restoration sites. It's very arid in the Angeles, gets minimal amounts of rain. And so container plants were doing really well. And then we started to see a lot of things starting to die. And it had been a fairly rainy year. All of these plants were irrigated, and we started questioning what is going on. And I happened to see a presentation by Susan Frankel where she had started talking about this pathogen called Phytophthora. And so I started wondering, I wonder if this could be a problem. And just that nagging feeling in the back of your head, I should probably look into this a little bit more. And so we called her up and asked her, hey, can we maybe try doing some sampling? So let me tell you a little bit about what Phytophthora is, other than a very hard to spell word. It is a water mold. The name in Greek means plant destroyer, so it is not very hospitable to plants. I don't know what the hell it is. It's like this conglomeration of fungus and water mold and algae. I don't try to really understand that. It mostly really spreads in water, which you would think, why in Southern California would this be doing well? So I was like, we're gonna be fine, but we'll just check it out and see maybe if this is what it is. But it's really a pathogen that mostly affects agricultural fields. So that was another reason I was like, we're gonna be okay, it's great. But really what was kind of making me think about it was sudden oak death, which maybe some of you have heard, especially if you come from California. Phytophthora ramorum is what causes sudden oak death, and that is causing massive destruction of all oak species and tan oaks in California. So you can see over 50 million oaks and tan oaks have already been killed just from that one species of Phytophthora. There's Phytophthora cinnamomi, which also has killed, it is a plant destroyer, right? So this one was happened to be in a nursery where they were outplanting this rare Arctostaphylos manzanita, and then it just really decimated the entire population. So, after hearing these, I was, again, getting more and more nervous. So talking with Susan, we started to set up this testing that we were gonna do. So we did 2 rounds of testing. So testing of the restoration sites themselves, and also we, in the wings, had over 200,000 container plants that were waiting to go out into the field. So I wanted to do both at the same time. So testing of actual restoration sites and testing of those container plants that we had growing. So for the restoration sites, what you do, this is the weirdest experiment, it's like voodoo. So you collect soil from the plants that looked diseased or dead, and you stick a pear in it. Sounds so ridiculous. When I heard this, I was like, oh, this is exciting. So I put all these pears. My husband loved me. I had pears all over our house, in the closet, because they have to incubate for 4 days. So silly. And then for the container plants, what you do is you collect the effluent from the plants. So you stick these plants over a tarp, and then you get a cooler and you drill a hole into it. It's and then you float. You could see the little green pear floating in that. And the effluent, so you water it every, 30 minutes for 5 hours. And what's happening is the zoospheres of the Phytophthora are flowing into the water, and they are attaching to the pears, and they're forming lesions on the pears. And the more lesions you have, the more Phytophthora is in that water source or is in the plants. So we did this on a bunch of different sites and a bunch of different plants. So we tested 6 of our 15 restoration sites and, 15 sites, and 6 of them tested positive for Phytophthora in them. I was like, no! And then we tested all 4 of our nurseries, and every single one of them had Phytophthora, which if you were in the nursery trade probably isn't that surprising. Phytophthora is fairly common in nurseries. I was talking about the sudden oak death. Sudden oak death is known to have come from the nursery trade. That is where it started from. So I'm not here to like rag on nurseries. We obviously need nurseries. They're obviously a great thing. But it was just to bring awareness that this is maybe something that we need to think about, especially given these results that all of those nurseries had Phytophthora and multiple kinds of Phytophthora. And most kinds of Phytophthora that have been identified in the United States are thought to be non-native. This is something that researchers are really working on to find out, like do we have our own native Phytophthora? And that is still somewhat inconclusive. But the ones that are causing all of this damage are definitely non-native species. So we tested all these different species, and certain species are certainly going to be more susceptible to Phytophthora than others. So a lot of the shrub species, which is mostly what we were growing, those tend to be highly susceptible. So obviously the oaks I talked about, manzanita is also, there's certain aspects about them, I guess, that make them more susceptible to others. And one thing that's interesting in the nursery is that you can't always tell if something has Phytophthora issues or not. And there's can be multiple reasons for that. It's mostly a fungus or a water mold or whatever it is that attacks the root system, so that can be something that's difficult to notice. So just looking at the plant itself isn't going to necessarily tell you that you have Phytophthora, and that's why the effluent testing is really important to do. And the same with it out in the field. So like I said, we do understand the importance of nurseries, but there are a couple things that you could do that make your nursery less susceptible to having Phytophthora issues. And one of the key things is to not have plants on the ground. That's how Phytophthora spreads. So it's, water pools at the base of those plants and it moves to the next plant next to it and it gets taken up into that plant. So having plants spread out on benches, that really is your number one thing that you can do. Also, not reusing plots without sterilizing them in between, and solarizing your soil are some of the main things that can really help not spread Phytophthora around in a nursery. There is an accreditation program in this working group for Phytophoras. They have a whole list of best management practices, some of them fairly intense, on how to make a nursery more phytosanitary. And there's an accreditation program that's starting in California for nurseries to help them meet these BMPs. And then they could be accredited, and then when you're buying from that nursery, hey, this is a Phytophthora-free plants that we are picking up. So after learning all of this, the implications, we have these 200,000 plants waiting in the wings to go out. We had to make some really hard decisions about were we going to take some of those plants. And in the end, we ended up taking about half of them. There were species that just looked great. They were well separated from plants that had tested positive. So we were able to compromise, but it was very difficult to throw 100,000 plants away, right? Like that does not feel good to anybody. And it made us start thinking about, we really thought container plants were where we were moving, and we started thinking again more and more about doing seeding and what could we do to improve our seeding game. And so there were a couple things that really we started changing in our specs that helped us out to really achieve better success. And the first thing was to increase our seeding rate. So we were we were using all locally collected native seed. super expensive, so we were trying to get away with 80 seeds per square foot, and we eventually just bit the bullet and did 120 seeds per square foot, and that really helped us out, that amount of increase. We also, when we first started out, we're doing just like the regular one-step hydroseeding application, and that really doesn't work well, especially in arid environments. It needs to be at least a two-step, or what we moved to was a three-step hydroseeding method. And if you're not familiar with hydroseeding, what that means is to put your seed down. We typically like to do that by hand, then to rake that seed in to have a good seed to soil contact, so maybe 1/4 to 1/8 of an inch buried. And usually that was by hand, but sometimes you can use like a chain harrow behind an ATV. That works pretty well for most soil types. And then covering that with hydromulch on top of it. And so the hydromulch application was a wood fiber at about, I think for these particular sites, between 2,000 and 2,200 pounds of wood fiber mulch per acre. So, and the reason that, we went with hydromulch is because these areas are, you could tell, like on the side of the slope, you're not taking a drill seeder out there anytime soon. We knew that hand broadcasting alone in arid environments does not work well. We were seeing like maybe 1 to 5% germination rates maximum. It's really important to get some sort of cover on top of that. Another thing we did was concentrate more on workhorse species. At first we had these elaborate species lists that were like 40 to 50 species long, and we just weren't getting the quantities that we needed. So we narrowed that down for our seed collectors and really focused in on like what's doing the best. We're just gonna concentrate on those. We're gonna take a hit on biodiversity, but the alternative out here is to get stands of weeds. And we figured it was better to have that, like, take a hit on the diversity just to get natives in place. We also had relied entirely on forest collections, and we started to realize like this was starting to have implications of taking tons and tons of seed from the forest, so like thousands of pounds. And we decided we really need to move to the seed increase realm instead of hammering the forest over and over again with collection. And so particularly for grass species, we moved to working with growers to grow out our seed. We also, in the beginning, were like, this has to stay within the same tiny little watershed for our collections. And we started to move to like, okay, maybe it's good if you're just within the forest in general. Like if it's the same vegetation type, the Angeles has certain mountain ranges that are faces that are very different vegetation types. So we just stuck to like that side of the mountain and just called it good, because it was, again, we were having trouble getting enough seed if we limited that collection to really small areas. We also concentrated on our seed bed prep, making sure what we wrote in the specs. You can write whatever you want in specifications, but you really have to be out there with the operators, making sure that they understand what you wrote and that you're on the same page about implementing those specifications. So in particular, doing decompaction down to certain levels, and then making sure that that seed goes on top of those freshly, like the soil is fresh, and putting the seed right on top of it within 48 hours. And after the seed's put down, within 24 hours, putting the mulch on top of that. Otherwise that hard pan crust starts to form and you really see reduced germination rates. We really relied on container plants to block OHV access to a lot of these areas. Believe it or not, people try to drive across stuff like this. And so we really focused, instead of using container plants, on things like boulders and vertical mulch to screen those areas to try to keep people off, which for the most part worked fairly well. So just really quick, some practices because there can be some implications of bringing Phytophthora in. There's not a lot of research on this, but there is some concern of bringing Phytophthora in with your seed that you may collect. So these are a couple points I would say, working with Susan, that she has passed on to me. So like, you don't want to collect seed from the ground. That could be a way of importing Phytophthora in with your seeds. This one's kind of a no-brainer, don't collect seed from plants that look diseased. And for this mostly applies to species that are hammered, that can really be susceptible to Phytophthora, is to try to choose plants that look really healthy and robust. And a lot of times this is like when you're dealing with tree species that might have resistance. Those might be good species to bring along with you, knowing that Phytophthora is moving through the landscape. And that's it. There's a ton of people that really are, like I said, know what they're talking about when it comes to Phytophthora, and me not being one of them, and these are the folks listed up here. So thank you for your time. Sure. Go ahead. Yeah, I know. It is the strangest thing, right? It really makes you feel like a mad scientist when you're like dipping pears and stuff. I don't know. Again, some crazy smart person came up with like, there's something about Phytophthora that causes lesions on the pears. And then what they do is they take a culture of that and they put it on an agar dish and they can watch the Phytophthora grow, and then they need to do genetic sequencing to figure out what the Phytophthora is. Sure.
Were you concerned about which species of Phytophthora was, or were you just any Phytophthora was like you threw it?
Yeah, no, that did factor into our threshold of which plants to take. I don't think we took anything that showed up positive for any Phytophthora, but certain ones, like if it was close to a really bad one, we're like, oh, we're not even going to take that. Yes, so certain species like Cinnamomum, Remorum, absolutely wouldn't have taken anything that showed up from those particular species. Sure.
So it seemed like there were outplanting Phytophthora symptoms previously, before the study started.
Yeah, that's...
Translocation of that
Yes.
Phytophthora outward into the wildlife.
Yes, thank you. I forgot to mention that. So the forest is still monitoring that, and I think we're on year 6 of monitoring that. We are not seeing it spread. It is still present, but we're not seeing it spread out of the site. Yes, at the planting sites where we found it. Yeah. Go ahead.
Who does the testing of the species?
Yeah, so in California, it's CDFA does that. Food and Ag will do that. And I think probably in other states it will as well. Yeah.
Given all the rain California's getting this year, is there concern that there could be like a big outbreak?
Yes. Yeah, so we're really interested to see how it's going to look this year after testing, for sure. Sure.
Is it mostly affecting woody species, or can it affect grasses and wildflowers and things too? And if not, like, why not?
You know, I don't know that we can answer that as well now. The species that really seem to be impacted are shrubs. The species that we tested happened to all be shrubs. We didn't have grasses or annuals that we were growing as containers, so it's hard to say. But it was brought over, like Remorum was brought over on sticky monkey flower, which is sort of like a sub-shrub, right? But I'm not sure. That's a good question.
In the nursery setting, like especially with trees and stuff, did y'all try any like fungicide drenches?
Yeah, so fungicide will suppress Phytophthora, but it will not kill it. And that's one thing when we were doing testing at nurseries is we required them to not use fungicide for 30 days prior to us doing testing. So yeah, the fungicide is a way of masking it, but so it can make it difficult to detect it. But then when you go out to plant it, it can come back for sure.
What was the time between testing to knowing what you had? Was that like a week or 2 months?
Oh, it took a while. I would say at least a couple months. I think 2 months. Yeah, it takes, because it's all this genetic work, right, to get to species. We could know a lot sooner if there was Phytophthora or not. And there's more and more. We did try, like, there's Phytophthora detecting strips that you can stick in the soil. They just weren't as reliable as this effluent testing was. Yeah.
Can you do the certification programs? Is it just those BMPs, or is it also testing for?
Yes, it's both. Yeah, to be accredited.
Accredited.
Yes. Yeah, for the BMPs, I think it probably talks about the protocol for testing in there.
Testing is part of the BMP process. So
Yeah. Sure.
I just want to say kudos for actually saying that you have this. A lot of nurseries, I think, do have this issue, and they keep it very under wraps. So,
Oh yeah, yeah.
kudos to you for actually bringing it up. So, you know.
Yeah, it's just something we need to learn to work with, right? And maybe it's okay in the ornamental trade. I'm sure it's in everyone's backyard, right? But for native restoration, and particularly in California, it's caused a lot of havoc. There's been some testing for Phytophthora in Oregon and Washington, and they really haven't found much of it.
I'm in Oregon at a nursery,
Oh.
and we're right on the edge of sod country in the Squaw Valley, and we're absolutely terrified. Like when I grow tan oak at my nursery, it's a whole scene when we bring those in, like the sterilization type
Oh, good.
we go through. So it's definitely on everyone's mind because it affects viburnums, Doug fir, and like root grapes and Doug fir. That's all we do.
Yeah, setting up a testing kit is like $30. It's time consuming to run, but it's pretty cheap to set it up. Thank you.